Today, like when I was talking to Lionel about the talk, I thought it would be a good idea to give a talk um, on sort of more of a historical aspect and how the whole metabolic optical imaging came in space with in terms of phasor now and originally ratiometric measurement. So the whole talk is mostly focused on sort of aspects of metabolic imaging that you can do and how they're correlated with more traditional sort of measurements and where we are now. And part of that newer information, Alex already talked about, so I probably won't go into as much detail anyway because he is the one who knows way more about it than I do. Right. So going back, the first papers that sort of started the field is a science paper that came out in 1962 from Beaton Chance, who was in charge of the, I think, University of Pennsylvania at the Metabolic Imaging Lab. And the first paper was this paper called Intercellular Oxidation and Reduction in Vivo. And what they did is basically they looked at mouse where they had these windows in brain and in kidney. And by looking at, it's not an imaging measurement, it's a spectroscopic measurement. And they were looking at the emission spectra and the intensity in these systems where you diffuse different gases, oxygen and nitrogen through it, and what happens to the system. So DPNH here is basically NADH. It's a different way of saying it. But so this is the NADH spectra. And what they saw is in a suspension, if you take the cortex and you suspend it, in aerobic anostic transition, there's a shift in this spectra. So the spectra shift. So that was one of the main measurements. Another thing was, well, if you, both in kidney and brain, if you pass oxygen and then if you pass nitrogen, what you see is because of the metabolic changes that is happening due to the changes in the environment, you'd see intensity increasing and decreasing in the NADH sort of signal. It would increase and decrease. So that gives an idea that, well, these sort of measurements based on optical properties of these molecules can probably be used for looking at metabolic changes. So this is both in kidney and brain. And you can see there's a ventilation period, and then the start signal, it goes up, the intensity. Then in 2002, when Watoev's lab published the two-photon spectroscopy and microscopy of NADPH and flavoprotein, that sort of expanded this a lot more. The main reason is all these measurements, they're very much UV excited. So if you're thinking of cells and tissues, they don't always like the energy that is coming from the ultraviolet radiation. So they burn. And however, if you can use two-photon, you are not exciting all the molecules, you're only exciting part of the system, only at the focal point, so you could use it. And if you look at carefully here, like either at 740 or 720 nanometer, where you have excitation of both FAD and NADH and NADPH, the specters match, you can excite both of these molecules, which is inside the range of a common titanium sapphire two-photon excitation laser. So that's what sort of started using. And these are the two-photon spectra, which means by using different spectral windows, you could separate out the NADH spectra from the FAD spectra and possibly use them to look at metabolic changes. So the first sort of idea is, well, what can you see and what we can do with it? So this is a very simple diagram of the energy cycle in a cell. So you have your TCA cycle, you have your glycolysis outside. Two of the major component of this is the coenzymes NADH, which is transitioning from each other in different steps of this process, and FAD to FADH2, which is again a coenzyme. Now, FAD and NADH, they are bright and fluorescent, so you could see them. FADH2 is not, and NAD plus is not. They're dark, so you can't see them. So the idea, well, in terms of fluorescence measurement, then is, well, can we use the NADH and FAD autofluorescence? And can we use them So, and they are already present. So these are intrinsic. You don't have to add anything to the system. So you can look at tissues and cells and even animals. And the idea again is, can we use them to look at metabolic changes? So one of the fast measurements was basically a ratio of the intensity in one channel by either normalization of that intensity or by one of these two variants, right? So there, in literature, there is a intensity of FAD by the total intensity, or FAD by NADH, or NADH by FAD, or NADH by FAD plus NADH. So all of those have been used in the past. And the idea there is this sort of ratio is correlated to the NAD plus to NAD plus and NAD. So NAD plus to total NAD ratio, and that's known to be a marker for the metabolic changes. So if we can correlate that. Now, again, FAD and NADH can be separated spectrally. So this is rather simple of the experiment. You could use 2-photon excitation to excite all 4 of them, and you can see them in different emission range to calculate this. Now, in terms of validation, well, whenever you are proposing a new technique, you have to validate with more standardized technique. So one of the fast measurements was comparison with liquid crystallography mass spectroscopy, which is just looking at the individual molecules and then calculating the ratios. So if you do that, if you look at LCMS of NAD plus to this ratio that one mentioned in the, and the FAD to NAD, you see a very high degree of correlation, which means you're seeing basically the same thing. Now, one of the things that any of this LCMS or even techniques like CFARS, which will come in a second, cannot do is it cannot tell you what's the diversity of a system, what's the heterogeneity of a system. So if you look at human stem cells and bone marrow derived cells under MSC propagations, osteogenic or adipogenic differentiation, you see a difference in the metabolic, and it's overall there is a difference, but also there's a huge heterogeneity in this system that you can see that you couldn't see otherwise. Another experiment to look at is also, looking at precancerous epithelial tissues. So all of these measurements are more sort of, it has expanded a lot more in cancer research because of the difference in the metabolic changes happening in the cancer cells compared to normal cells, going into the Warburg effect. So that's sort of the reason that it has been expanded there more, because it's a larger differentiation in here. But if you look at, again, the LCMS versus optical radius ratio, you see a very high degree of correlation. So you see the similar thing. And like I mentioned, those LCMS measurements, you don't have inter and intracellular variability, but which you can see because it's an imaging technique, optical imaging technique to start with. Now, another validation is with optical oxygen consumption rate, which is the seahorse assay. So the idea here is, well, you have your cell membrane, mitochondrial inner and outer membrane, and you can actually block different parts of this metabolic cycle. You can add oligomycin, which inhibits the mitochondrial synthase. You could add FCCP, which is gonna stop basically mitochondrial consumption of NADH by uncoupling the electron transport from the ATP generation. And then you have ortidronine, antimyosin A, which basically stops complex 1 and complex A. So all of these changes would affect your metabolic behavior. And if you look at this data coming from the optical redox ratio versus the NOCR, you see exactly the same trend as you see in both. The higher error bar here is obviously because you're averaging multiple cells, while you have individual sort of information from each cell. Here you are only averaging the whole dish, so you don't have that information. So obviously the data looks cleaner, but it's actually, this is more representative of the diversity in the system. So again, you can look at that redox ratio as a color map, and you can see that changes happening in the metabolism and sort of real time. Again, another sort of work is if you have different types of cancer cells, and if you added normoxia or hypoxia, if you look at optical redox ratio and in OCR, you do see exactly what happens from normoxia to hypoxia in each case, and they're completely correlated. And if you look at the metastatic potential, so you are looking at post-hypoxial change in redox ratio, the percentage change versus metastatic potential, you see as the cells are supposed to be metastatic are highly metastatic. So there's, this is still in 2016, so it's a few years old, and that sort of starts adding more information and heterogeneity to the system that you can see. Now, the question is, well, this works, so why do we do lifetime experiments? Because you heard from Enrico, you heard from Alex, probably heard from Dave earlier about phasor and other stuff. So, well, why do we have to do lifetime? Now, the intensity is, well, it's correlated with the quantum yield. If you have multiple species in the same spectral window, if one species has a higher quantum yield, it would, and they have similar mole fraction, which is shown in Chi, they would actually attribute more fluorescence intensity. So intensity is always defined by that. So, and if you look at here, if you look at NADH, which has a very low quantum yield for free, and a much higher quantum yield, it's a calculated quantum yield from the bound, you have a little bit of bound NADH is going to contribute a lot more to this system than a lot of free NADH. Right? So, but if we can do lifetime, and I don't have to go into the lifetime, so you have absorbance and fluorescence and the time that it spends in the excitation, which is dependent on the all sort of behavior. The molecular structure is one, environment is another one, and you actually can look at the lifetime. The good thing is it's independent of concentration, so you could potentially separate out these species, free bound NADH, free and bound FAD, and can we use them for optical metabolic imaging? And then you are not dependent on this, so you can look at multiple different systems, even though their total intensity is decreasing. So you can possibly even increase that by adding more laser power to it. So again, the first paper to use it was Damien Bird and Nirmala Ramanujam's lab. I shouldn't make that up, but it's in looking at MCF10A human breast cancer cell levels, and with looking at in NADH, right? So these are they are under differential cell confluence. So as you increase the cell confluence, you see the free and the difference between free and protein bound. But the thing I want you to focus on is the ratio of the free to bound. You see the ratio of free to bound, the one in the line goes up pretty drastic as the confluence goes. However, if the cells are serum starved, so their metabolism is changed, this increase is very, very low. And if you look at the protein bound under potassium cyanide, which is a guy which would induce glycolysis, you see very similar behavior as that of serum starved. So these tells us, well, we can use this sort of method to look at glycolytic and oxidative phosphorylation changes in a system. And one of the first important paper was from Melissa Scala, when they looked at cytosolic NADH that with, well, it's basically in precancerous epithelial cells. And so this is your intensity ratio, optical redox ratio, as I mentioned before. This is the average lifetime. And as you see from normal A and D to as you are going low grade and high grade precancerous tissues, and is going in depth because there's a less availability of metabolic oxygen. You see the average lifetime going down. You also see the redox ratio goes down. So these two systems are now completely correlated. And again, this is the ratio that is very important between the protein bound and protein free. And you see this ratio in terms of bound is going down as you're getting more and more glycolytic. So this gives us, again, a way to look at this system. Another quick example is, well, looking at the same system. This is in the brain where they have a way to continuously add gravity-fed topical reagent, where they are adding control deoxyribonucleic glucose, iodiacetic acid, which would inhibit glycolysis, fluoroacetane and arsenite salts of sodium, which would inhibit TCA cycle, rotenone and potassium cyanide, which would inhibit electron chain, FCCP, oxidative phosphorylation, and bicalculated, which is their focus is on model because that's what the paper is based on. But you see in every case, you see the changes in metabolism inside the brain as you are adding this system. So that also tells us, well, It's a continuous behavior that is continuously changing, which means we can monitor it in a live animal. This is done in live animal. So comparison to with OCR, the idea again here is if you're free to bound NADH, it goes up, your redox ratio goes down, and your OCR, the oxygen consumption rate that is measured from the seahorse assay, would go down. So if you look at mitochondrial stress test, so compared to control, if you add oligomycin, your free to bound ratio goes up, your OCR goes down, which is expected. The reverse is true in FCCP. And if you do the glycolytic stress test, which is looking at systems which is either by adding glucose or oligomycin or 2-DC, every time you have an increase in this, you have a decrease in that, increase in, again, free to bound NADH, decrease in the OCR. So all these results show that we can use the NADH lifetime to do this sort of analysis, and only based on NADH lifetime we don't. Now, I focus so far on this sort of behavior. I was not sure completely of the schedule. So obviously from Enrico's lab, Kate Pate and Pierre Stringari, their work on wind signaling has basically the exact same behavior explained in terms of phasor diagrams. Okay, so coming back to phasor, you have all heard this, so this is going to be very short sort of explanation. So you have your lifetime, these are calculated lifetimes of different ones, and you have your sine and cosine curve. So if you have a very short decay, you do the calculations basically by it's a multiplication at this point, and you get a very you get something in a G close to 1, S close to 0, and as you are increasing your lifetime, they move along this axis. If they're mono-exponential, they will always appear along this universal semicircle. And if they're multi-exponential, they would appear, a sum of exponential, they would appear inside. So that's just a quick calculation to show based on those lifetime where they appear. One of the major important property, and one of the reason I think phasor is so well used, is reciprocity principle, which means if, let's say we have an image of a cell, and we calculate their fluorescence life or phasor lifetime, and we get these different regions. Well, first, of course, you can select this region, and you can see where these regions are, what these lifetimes or phasor clouds are in terms of the actual image. However, you can also select out different parts of the image, and you can start seeing where those points lie in a phasor plot. So this back and forth between going from the image space or phasor space in terms of phasor colored image to the actual image space and calculating their phasor point is what gives the phasor is very high sort of applicability in imaging, especially metabolic changes, in small changes in different parts of the behavior. Another thing that Alex mentioned is the linear combination. So this is just shown in terms of solution. So let's say we have a solution A and B. Our solutions would, phasor points would lie in these 2 points. However, if you mix those two, which you think, if you think about that in terms of image space, is basically the same behavior if you have single pixel which has 2 colors, right? 2 different dyes. It's the same as almost like having a solution of 2 different dyes. And then it would lie somewhere in between this line where the amount of the distance for each case would depend on their fractional intensity, the amount of intensity coming from each, and is reciprocally related to that. So, but that gives us an opportunity to use this and go back and see what's the actual fractional intensities are. So in terms of metabolic quantification using phasor approach, you have our free NADH where they're collapsed on top of each other, the 2 rings up here. And I can show it easily in the protein bound structure where you have this ring and this ring, where the adenine and so things are on top of each other. They're basically collapsed and they have a quenching effect. So their fluorescence lifetime is low. When they bound to protein, they become large. They go away from each other, and that increases their lifetime. And if you look at the lifetime in terms of this bound to LDH, lactate dehydrogenase, this lies up here, this one is up here, And these are just simulated data where, not calculated data, not simulated data, where we are adding different percentage of the one to the other, and you see this shift going from one to the other. Now, just keep an eye on the 5% and 10% of the, because its intensity is lower, you see a very little shift when it's a little change in the protein bound NADH. However, in phasor term, that's different, because once you calculate the phasor positions of these decays, you see a much larger change in the phasor space, because in phasor, this is a tangential, this is a tangential scale. So changes here, this is 0 to 0.4. Here it would be 2 to 3.4. So it's a very small shift on this direction, very large shift on this direction. So if you are looking at especially things which are changing a small amount, especially by addition of the brighter species, you see a large shift in this direction. And this line between the free and bound NADH is called the metabolic trajectory. And so shift along this trajectory gives us metabolic changes. And then we can use this to actually convert them back to fractional intensity of the free NADH, basically based on these positions. And we can see quantitatively between sample to sample and between, let's say, in tissues, animal to animal, what sort of behavior we are getting. So the first work sort of showing this in terms of metabolic in phasor was Kiera's paper, where if you add reducing condition by potassium cyanide, it would shift towards more free NADH. If you add oxidizing condition, hydrogen peroxide, it would move towards more bound NADH. And this can be seen from the color map here. She always used free NADH as a more purple color, so you see that sort of behavior. And if you do another experiment where you actually add high glucose or sodium glychloroacetate, which is sort of a not direct measurements, is actually changing the glycolysis oxidative phosphorylation. And you could see the glycolysis has a similar behavior and oxidative phosphorylation has the opposite behavior. So we can use this now to look at changes of this system. Now, how do we do this analysis? So this is from our paper with Leonid. And so we do the image acquisition and then we calibrate it. And the way the calibration works is mostly we use a fluorescent dye where you get some point of this, but first you correct for the angle and then you force it back to the semicircle, because these dyes are usually mono-exponential and you know what the correction factors needed. More traditionally, for people who use scattering, you can do the same. You can basically position it back to this position, which is a lifetime of 0, which is the scattering lifetime position. Once you have that, once we know the referencing, we reference the sample. We mask for looking at different areas. We can do image filtering, which doesn't change the actual image resolution, but it modifies the pixel resolution, and because it's a 3x3 algorithm on the phasor space. Or 5x5. And then you can select the intensity, and then you can analyze for biological fluorescence, rate, concentration, pH. And in terms of biological fluorescence, is either you can select image, average GNS, which sort of mentioned 2-component, which is sort of showed in the last one, and then the 3-component measurements, which is Alex sort of mentioned, and I'll go back again in a second. Why do we do? It's a feed-free, so it's very fast. It's visual in nature. It decays the phasor properties assigned to molecular species and not lifetime. So we're not calculating just lifetime without knowing what the species is. We are actually assigning to the specific species and you're calculating their fractions. It's an image, so all the image analysis procedure can be applied to the phasor space. And then the filtering without changing the resolution of the image. So this shows a filtering effect of 3x3 and 5x5. And if you do that and you basically add that to the center stage, you actually, the image itself doesn't get blurred, which is a big addition in sense of this analysis. One of the things from Rupsa, who was a postdoc, is metabolic reprogramming in astrocytes. So they are looking at Huntington diseases and how the disease progress, right? So the idea is it would go to switch to fatty acid oxidation, and it's getting loss, which would create energy benefit loss, but it would be loss damage. Okay. So if you look at different areas of brain toxicity, in areas which is susceptible, the STR, the striatum, or the resistant, which is the cerebellum, you expect different behavior. And 20 weeks versus 80 weeks. So as the mouse is getting older and their Huntington is progressing, you see a shift in the metabolic behavior between the striatum, and the shift in the striatum is much higher compared to shift in the cerebellum. And you can actually sort of calculate that and see what's happening between aged and young mice with Huntington disease. So this is a collaboration that Indico had with University of Berkeley. One of the projects that I used is looking at spatial dynamics of SAT1, which dictates the metabolic transition in the cell nucleus. And the idea here is the gene plasticity. So when you're talking about plasticity, you have writers, readers, and erasers. These are the systems that actually modifies the how the expressions would happen. And you have to have a writer which will start the writing, you have to have reader which would understand it, and then eraser would take out the system. So SAT1, which is a deacetylase, is a de-eraser, and for it to be active, it has to bound to chromatin. So that we thought that we could look out through diffusion, the binding of the chromatin with the SAT1. And we used a method that we developed, which is called diffusion phasor. It's a way of using phasor to look at diffusion by pixel to pixel. And this is just simulated data showing different sort of diffusion, fast to slow, and you can actually select them out through converting them to the phasor space. Now, the idea here is, well, NAD plus metabolism is compartmentalized, and we're talking a nucleus here. So nucleus is part of this salvage cycle where you have different enzymes which converts NAD to NAM, NMN, and NAM, and NMPT, which is the actually the step which controls all of this behavior. So if we can actually either by modifying the SAR, if we can actually change that metabolic behavior, can we see it? Or if we can modify the metabolic behavior, and can we look back at the SAT1 activity? So that's exactly how can we study NAD metabolism in nucleus, and how can correlate with SAT1 dynamics. So we looked it through the phasor and FLIM of NADH, and the idea again is SAT1, when it's bound, is active, when it's freely rotating is inactive, and looking at that we should be able to see. And the way we did it, we control SAT1 activity by either mutating or adding a drug called EX527, which blocks SAT1 activity. We also block this salvage pathway by adding a dye which blocks this enzyme NMPT, which are our direct control, and then our metabolic control is addition of NAD plus and NAM to force this go back one way or other. So if we look at FLIM data, we look at wild type versus SAT1, and if you look at the metabolic imaging, the more red here is more bound. If you look at that between wild type and SAT1, you see a shift, SAT1 negative cells, while SAT6 negative cells, which should not matter, doesn't do anything. And then if you add the EX527, you see a timing-wise shift in the metabolic behavior with SAT6, with this blocking SAT1. So we can actually control the metabolic behavior by blocking SAT1. But if you add that to SAT1 negative cells, nothing changes, which is also expected. So we looked at all these fractions. Now we can correlate this with diffusion data. So if you block the SAT1 activity by either genetic manipulations or by adding this these purple colors, which are the binding and slower diffusion, like you can see here, they go away by both as a function of time in terms of the adding the drug, and here is obviously it doesn't have active SAT1, so that's why it's stayed up that way. Now we can look at that through the indirect inactivation. So again, we block these different pathways, and we can see with NAM, you see a more oxidative phosphorylation. But in negative cells, SAT1 negative cells, you don't see anything. In NAD plus, if you add, you first go up in terms of activation, then you go down, come back to the non-significant level. In terms of FK866, which blocks this salvage pathway enzyme here, you see more and more with time after treatment. And with untreated in SAT1 negative cells, you don't see any of this behavior. Now, if we look at the diffusion coefficient of that, we can add the FK866, and you see the same behavior here correlated to this as you are decreasing the SAT1 activity. You are actually decreasing SAT1 activity here, and you see the changes in metabolism. On the other hand, if you first block it and then add NAD to NMN, so you are sort of making it more active again. As you can see, there's a little bit more active compared to here, not all the way up. You can start seeing the activity coming back. So this way we showed how SAT1, which is a clock enzyme, can actually, you can activate, inactivate it by adding energy and other way around, right? So that's sort of what we did. Now, a word of caution is all of this phasor space depends on the proper fitting and transformation, because in literature you see NADH positions all over the place, especially bound one. And part of that is correlated to what enzyme it is binding, but part of that is also transformation from the, because most people don't take the data with FLIM box. Most people take the data with time correlated single photon counting instruments, and conversion of that can be tricky. So if you look at, and that's why you get all sort of behavior in terms of lifetime here, as I mentioned. And this is just a decay of the cumarin. Most people would use the decay to analyze is this black window here, which is the rise and the fall time, but the decay doesn't decrease all the way to 0. It's also a non-repeating signal, and phasor is a Fourier transformation, so this doesn't transform correctly. However, Enrico might have mentioned this. However, if you choose a window in between two consecutive decay sets where you have a repeating signal, you could actually transform it properly. And if you do that, you basically get this sort of behavior where it's roughly 3.4 to 4 nanoseconds is where our bound NADH is, and our free NADH is as always at 0.4, and that's the proper metabolic trajectory. And if you do that, this is showing the same data. Baker-Hickel gain of 2 is basically looking at this large area, and gain of 5 is looking at the small window. And with the small window, we get, depending on what we use for actually looking at, it gives us long data, and you can see in terms of cell, you sometimes get a differential behavior if you don't calculate the proper positions of the NADH based on the system that you are using, because right now here is outside the behavior, outside the window. Right? So what about fixation? Does it change anything? So there's a paper from Jane, who was a postdoc with Michelle and back in the time, and they looked at that. They looked at 2 different cell lines. It's a triple negative breast cancer cell line, and MCF tail, which 10A, which is non-cancerous epithelial cell line. And if you look at live and fixed in terms of mean lifetime in NADH channel, yes, the lifetime change. However, the behavior keeps the same. You see a decrease in 231 in the average lifetime. In live tissue, live cells, you see the same in fixed cells. And the mean lifetime, the behavior of that with in terms of sodium cyanide network, deoxyglucose, the metabolic pathway changes in the things that we talked about, you actually see exactly same behavior as in live and fixed cell qualitatively. So you can still use this on a fixed sample with paraformaldehyde or NPF. And this is looking at tissues. This is cells and live and fixed. You have the intensity histogram. They are highly correlated between cells. In terms of phasor, you do see a little shift but again, it doesn't matter if you're looking at correlative behavior between that system, as long as the samples are done in the same way, fixed the same way, you can look at that. This is cancer tissues by looking at NADPH lifetime, and you can see in normal tissue to cancer tissue in fixed sample, you see this shift in the lifetime, which is what you expect is a shorter lifetime, and which is expected also in terms of but caution again, this doesn't work for FAD if you actually are looking at FAD imaging and you have fixed tissue, FAD changes based on behavior of FAD changes based on the fixation agent. So the final part, quick, in case I'm running a little late, is oxidative stress. So can we see something else other than NADH and look at other stuff? So this is metabolic. This is Rup Phasor, who is a staff scientist at University of Wisconsin now. And she was looking at with oleic acid fed cells, and what she saw is if you add oleic acid, you see a secondary behavior and you can map it out. They're coming at different places. And she did other imaging techniques to correlate this. She did coherence anti-Raman scattering, which looks at droplets, lipid droplets, third harmonic, which looks at the difference between the metabolic areas, and you can look at that by comparing the third harmonic to see the place where you have this long lifetime is a droplet. So that's sort of, it's telling you it's coming from the lipids, right? Now you can look at that through the normalized, so you can also look at through Raman shaped, where they showed that it correlates with wherever you have triglycerides. So this is the triglyceride signature, this particular energy lane, and it correlates with this presence of these lipid droplets, which we think are basically breakdown products from oxidation of lipids. And she looked at through metabolic imaging in terms of normoxia and hypoxia in chambers, and you can see with oxidative stress, you see more and more of this behavior. Now that's great. We do need, but this is sort of a quantification by putting points, and this is sort of what I'm going to tell you is the first quantification we did, and Alex has obviously changed it a lot and improved it over the time. So in terms of how it should look, if you have only metabolic trajectory between free and bound NADH, your phasor is in that line, along the line of the metabolic trajectory. However, if you have a second, or if you have a third component, a long lifetime species, or this oxidized lipid species here, depending on how much it is, it will start dragging it towards it, right? It would always be within that within that triangle. This is again just to show calculated data of the three independent species, 8, 3.4, and 0.4, and differential version of this by adding differential amount of each of them. And if you look at them, they are always within that that system created by these three phasor points. So the first analysis we did, what we did is basically it's a projection based algorithm. You take a line, you go through an image point, and you go back to the line, which is the metabolic trajectory. Now, the distance between where it catches, so the projection of this phasor on this is basically your metabolic behavior along the metabolic trajectory. If you think about it, if you didn't have this component, that's what it would be. And if you have this component, it's shifting it, so you're pushing it back. So that's the metabolic trajectory. And how close this is, close to this rate, tells you how much of this fraction of this rate is. So we looked at FLIM images from different liver. This is the first sort of data with Western diet, which is high fat, high cholesterol, low fat diet. This is the image of the low fat diet, just color coded according to this. We didn't have the 3 color coding that Alex mentioned yet. And this is just by selecting that really long lifetime species, and you see the behavior of all these fat droplets all over the image in a high fat diet mouse, which are fat, and you can see that. And we can actually correlate these distributions and to show that, well, in a high fat diet, you see more and more long lifetime species. Resolution of 4 components in the pixel. I'm completely going to skip this blind component. I'm completely going to skip this because Alex just gave a talk on this. So this is all his work.